Hi guys, how are you all? I hope every one of you are super cool, super good. And today I'm myself Saranya, your educator, in your channel Gurmantra Shikshaka Granth, going to teach you TCA cycle. Okay, so TCA cycle can also be called as tricarboxylic acid cycle. This is very important pathway. Basically, glycolysis, TCA cycle, and electron transport chain. These three are like a linked reactions. All these three things happen one by one, step after step, which are really important for us to study. And you might get a lot of questions from these pathways. So it is important that you cover these very well. Always remember the mantra, study, revise, practice and repeat. Okay, so let's go to the topic. So first we are going to talk about the link reaction between glycolysis and tricarboxylic acid cycle. Okay, so the link reaction is nothing but how is it going to link, what it is going to link the glycolysis to TCA cycle. Okay, that is what we are going to see. We actually, you know that glycolysis is a process that happens in the absence of oxygen. It does not require oxygen. That's why we call it as anaerobic process also. Okay, we can call it as anaerobic process also because it does not require any oxygen. But most of our cells, most of the organisms survive in the presence of oxygen, right? Most of the organisms survive in the presence of oxygen. So after glycolysis, where we saw the first step starts with glucose, which is a 6 carbon compound, and it ends with pyruvate, which is a 3 carbon compound, right? This pyruvate undergoes oxidative reactions, which means it undergoes aerobic reactions, and it gets converted to a 2 carbon compound. We call it as acetyl CoA, so that acetyl CoA can enter into the tricarboxylic acid. Okay, pyruvate gets converted to a product that gets into the TCA cycle. That is the link reaction here. Okay, so you see the first stage of aerobic respiration is the link reaction, which transports pyruvate into the mitochondria. Why pyruvate has to be transported into mitochondria? Because glycolysis happens in your cytosol. Okay, so glycolysis happens in your cytosol, which means pyruvate will also be in the cytosol, which has to be transported into the mitochondrial matrix so that your tricarboxylic acid cycle will happen. The link reaction is named because it links the product of glycolysis with the aerobic process of the mitochondria. What is the aerobic process of mitochondria? It is TCA cycle, tricarboxylic acid cycle. Pyruvate is transported from the cytosol into the mitochondrial matrix by carrier proteins. You have carrier proteins which transports the pyruvate to the mitochondrial matrix. Okay, it is on the mitochondrial membrane that will be transported inside the mitochondria from the cytosol. So in the link reaction, what happens is the pyruvate loses carbon atom by decarboxylation, which means carbon dioxide is released. So this is the one step. Second step is, since it loses one carbon atom, it becomes a 2 carbon compound. We call it as acetyl group. Okay, it has an acetyl group. And this acetyl group loses one hydrogen atom via oxidation. And what is the oxidation happening? NAD plus is converted to NADH. Okay. So this reaction is a redox reaction where oxidation of NADH will lead to reduction of NADH. Okay. So first one is decarboxylation, which means carbon dioxide will be removed. Next is NADH will be produced. Third, to this acetyl group, coenzyme A will attach. Okay, will attach and form acetyl coenzyme A. Short form we can call it as acetyl CoA. So acetyl CoA will be produced. That is the 2 carbon compound. So I hope you understand that in link reaction 3 steps are happening. The first step is decarboxylation, which means carbon dioxide is removed. Second step, NADH is produced, and third step is coenzyme A combines with the acetyl group to form acetyl-CoA. Okay, so which is a 2-carbon compound. So pyruvate, which is a 3-carbon compound, from the cytosol travels to the mitochondrial matrix using the transporter proteins, and there pyruvate loses 1 carbon dioxide and NADH is produced. The product is getting combined with coenzyme A, and finally you're getting acetyl-CoA, which is a 2-carbon compound. Okay, so since glycolysis gives rise to 2 pyruvate, okay, so one glucose molecule can give you 2 pyruvate, right? So 2 pyruvate is producing. From that, 2 acetyl-CoA will be produced. Okay. From this step onwards, the TCA cycle, we are going to concentrate one acetyl-CoA only, but you will have to multiply the reaction into 2 because 2 acetyl-CoA is there. So the cycle will happen for 2 times. For one glucose molecule, the cycle will happen for 2 times because you have 2 acetyl-CoA. So now let's concentrate with one reaction, and finally we will multiply whatever we are getting into 2. Okay, so that is the simple way. So always remember, for one glucose molecule, 2 pyruvate is produced, therefore 2 acetyl-CoA will be produced, and therefore the TCA cycle will happen for 2 times. Okay, I hope you remember it. So this is the link reaction. Okay, pyruvate, which is in the cytosol, which is the end product of glycolysis, which is a 3-carbon compound, is transported into the mitochondrial matrix using the transporter proteins, where it undergoes decarboxylation, which means when carbon dioxide is removed, and NAD plus will be converted to NADH, and coenzyme A is attaching and forming acetyl-CoA, which is a 2-carbon compound. So 2 acetyl-CoA will be produced from 2 molecules of pyruvate. Okay, you understood, right? So this is the tricarboxylic acid cycle or citric acid cycle or Krebs cycle. Okay, why do we call it as tricarboxylic acid cycle or citric acid cycle? Because one of the intermediates in this cycle or the pathway is having 3 carboxylic acid group. So since we call it as tricarboxylic acid cycle, and the compound which is having 3 carboxylic acid is citrate, therefore we also call it as citric acid cycle. Okay, so this is happening in mitochondrial matrix. I am just repeating the area where it is happening, that is mitochondrial matrix, again and again, so that it goes and fixed in your mind. It goes and stores in your brain permanently, because these kind of questions they'll ask you, like where is glycolysis taking place, where is tricarboxylic acid taking place. These are some of the potential questions which they might ask you. Okay, because they've asked you these questions in several life science exams. So they will ask you in the gate also. So these are some of the very minute details which we might ignore, but the examiner will definitely ask you these questions. So remember this, I'm just repeating it again and again so that it stores in your mind. TCA cycle happens in the mitochondrial matrix. Glycolysis happens in the cytosol. Okay. Okay, so first, for the tricarboxylic acid cycle, the link reaction we saw, pyruvate is getting converted to acetyl-CoA, and what are happening in that process? Carbon dioxide is released, NADH is produced, okay, and coenzyme A is coming in, and acetyl group will react with coenzyme A and form acetyl-CoA, which is a 2-carbon molecule. Remember, acetyl-CoA is a 2-carbon molecule. You can get that from fatty acids or amino acids also. It is not just useful for tricarboxylic acid cycle, it is also involved in fatty acids and amino acid cycle. Okay, so this acetyl-CoA, okay, which is a 2-carbon compound, interacts with oxaloacetate. Oxaloacetate is a 4-carbon compound. So 4-carbon compound oxaloacetate interacts with acetyl-CoA, which is a 2-carbon compound, to give rise to citrate. citrate, okay, which is a 6-carbon compound, because acetyl CoA here is 2 carbon, oxalate acetate is 4 carbon, so together they form a 6-carbon compound. We call it as citrate. Okay, acetyl CoA plus oxaloacetate gives citrate. Citrate is the compound which is having 3 carboxylic acid group. Therefore, the name of the pathway tricarboxylic acid cycle, citric acid cycle, because this compound is having 3 carboxylic acid group. So from citrate you will get isocitrate. In this picture, let's learn the name of the intermediates in the tricarboxylic acid cycle. I will teach you every single step of this, like what are the enzymes involved, what are the cofactors involved, everything I'll teach you. But first let's learn the name of the intermediates. Okay, so you know it is starting from acetyl CoA, which interacts with oxaloacetate to form citrate. From citrate you will get isocitrate. From isocitrate you will get alpha ketoglutarate. Citrate and isocitrate are 6-carbon molecule. From isocitrate, one carbon dioxide is removed, one NADH is formed, and it gives rise to alpha ketoglutarate. Also we call it as alpha Kg. Okay, alpha ketoglutarate, which is a 5-carbon molecule. From here, one carbon dioxide is removed and one NADH is formed. It becomes succinyl CoA, okay, which is a 4-carbon molecule. From here, it's not ATP, it's GTP. One GTP will be removed and it forms succinate. From succinate, one FADH2 will be removed and it will form fumarate. From fumarate to malate, from malate again to oxaloacetate, one NADH will be removed. So this is the reaction pathway, which is an 8-step cycle. First, I will make you remember, I'll give you practice of all these things. I'll help you, by heart, this tricarboxylic acid cycle. But firstly, let's remember the names of the intermediates. There is no trick. Actually, I'll give you a trick, but you will have to remember the names of the intermediates, because there are few things in your studies which you'll have to remember. There is no understanding. Understanding is to some extent, but the names of the intermediates, all these things you'll have to remember. Okay. So I want you to remember the name of these tricarboxylic acid cycle intermediates in this picture. So it starts from acetyl CoA, which interacts with oxaloacetate to give you citrate. Citrate will get converted to isocitrate, from isocitrate to alpha ketoglutarate, from alpha ketoglutarate to succinyl CoA, from succinyl CoA to succinate, succinate to fumarate, fumarate to malate, malate to again oxaloacetate. And the trick is, in the between I have actually given here the trick. This is not my trick, but this is the trick which is used widely, so you can also use it. Oh, can I keep studying science for med school? Okay, so the O for oxaloacetate, can for citrate, I for isocitrate, keep for alpha ketoglutarate, studying succinyl CoA, science, succinate for fumarate, med school, malate. Okay, so O, can I keep studying science for med school is the order, and you can remember the intermediates of tricarboxylic acid cycles this way. Okay, so the tricarboxylic acid cycle, or we call it as TCA cycle, also known as citric acid cycle or Krebs cycle, is major and energy producing pathway in living bodies. Okay, cells obtain ATP from the breakdown of glucose in the absence of oxygen, that is what we call as glycolysis. However, most organisms normally are aerobic, okay, and oxidize their organic fuels to completely carbon dioxide and water. And this cycle, TCA cycle, happens in the mitochondrial matrix. So now we are going to see the Krebs cycle. This picture I've given you with the structure so that you can understand how is the transformation taking place. So this is acetyl CoA, which is a 2-carbon compound, reacts with oxaloacetate, which is a 4-carbon compound, to give citrate. Citrate is a 6 carbon compound. Okay. You can see 1, 2, 3, 4, 5, 6. 6 carbon compound, and this is the speciality of tricarboxylic acid cycle because this compound is having 3 carboxylic acid groups. Therefore, the name comes from this tricarboxylic acid cycle or citric acid cycle. Okay, and this citrate loses one water molecule to form isocitrate. I mean cis-aconitate and isocitrate. Okay, this step is not necessary. Just remember from citrate to isocitrate it is going. From isocitrate, which is a 6 carbon compound, it is then getting converted into 5 carbon compound by release of carbon dioxide and NADH formation, alpha ketoglutarate. From alpha ketoglutarate, it becomes succinyl CoA. By release of one carbon dioxide and one NADH will be produced succinyl CoA to succinate. One GTP will be produced. Succinate to fumarate. One FADH2 will be produced. See, all these steps are continuously happening. From isocitrate to alpha Kg, one NADH carbon dioxide released. From alpha Kg to succinate, one carbon dioxide NADH released. From succinyl CoA to succinate, GTP released. From succinate to fumarate, FADH2 released. So you can see it is happening continuously. From fumarate to malate, malate to oxaloacetate, also one NADH will be produced. So totally in this TCA cycle, 3 NADH, 1 FADH2, 1 GTP and 2 carbon dioxide will be produced per acetyl CoA molecule. Since 2 acetyl CoA is there, this number of products formed should be multiplied by 2. Okay, in the Krebs cycle will happen 2 times. Okay, so this is the master slide or final slide where we are going to see with the enzymes. Enzymes are very important because only they carry out the reaction, right? They help you move forward in the reaction. So enzymes we are going to study. Actually, in the previous pictures, I've made you learn the intermediates. What are the substrates used in this tricarboxylic acid cycle and what are the cofactors and what are the reactions happening. Now we are going to see it with enzymes. Okay. So acetyl CoA, 2 carbon compound reacts with oxaloacetate in the presence of citrate synthase. Since citrate is forming, we call it as citrate synthase, synthesis of citrate. So the enzyme is citrate synthase, you will getting citrate, which is a 6 carbon molecule, 3 carbon here, 3 carbon here, also 3 carboxylic acid groups, so tricarboxylic acid or citric acid cycle. From this citrate to isocitrate, you are going to use the enzyme aconitase. Aconitase is the enzyme used to become isocitrate. Okay, this is isocitrate. Okay, this is the isocitrate. Here also it is 6 carbon molecule. From isocitrate, what happens? One carbon dioxide will be released, one NADH will be formed, and in the presence of isocitrate dehydrogenase. Dehydrogenase means NADH will be formed. Isocitrate dehydrogenase, you are getting alpha ketoglutarate. Okay, remember the substrate here and the enzyme is also named after the substrate. So isocitrate dehydrogenase, you are getting alpha Kg. Okay, alpha Kg is a 5 carbon molecule. From this also one carbon dioxide is released, one NADH is formed using the enzyme alpha ketoglutarate dehydrogenase. Alpha ketoglutarate, the name of the substrate, dehydrogenase, and you are getting succinyl CoA. Succinyl CoA you are getting as the product, and succinyl CoA is getting converted to succinate where one GTP is released and the enzyme is succinyl CoA synthetase. Since succinyl CoA is the substrate, the name of the enzyme also starts with succinyl CoA synthetase, means synthesis of succinate. Okay, from succinate to fumarate, your enzyme is succinate dehydrogenase and FADH2 will be produced. Succinate dehydrogenase, succinate name from the substrate. Then from fumarate to malate, fumarase. From malate to oxaloacetate, malate dehydrogenase, where NADH will be produced. Here you can see one common thing in the cycle is wherever NADH is produced, you are using the enzyme dehydrogenase, because here NADE plus is the cofactor. Okay, so citrate synthase, aconitase, isocitrate dehydrogenase, alpha ketoglutarate dehydrogenase, succinyl CoA synthetase, succinate dehydrogenase, fumarase, malate dehydrogenase. Okay, everything, the enzyme name starts with its substrate and then its function. Okay. So each molecule of acetyl-CoA entering the citric acid cycle eats the following. So you are going to get 2 carbon dioxide, 3 NADH, 1 FADH2 and 1 GTP. Since you are having 2 acetyl-CoA uh, since you are having 2 acetyl-CoA molecule, and the TCA cycle is also going to happen for 2 times, all of these products will be multiplied by 2. So 4 carbon dioxide, 6 NADH, 2 FADH2 and 2 GTP will be produced. Also you have to remember this fact that 1 NADH will eventually give 2.5 ATP, 1 FADH2 will give 1.5 ATP and 1 GTP is anyway converted to ATP only. So for one molecule of acetyl-CoA you are getting 10 ATP. 7.5 from NADH, 1.5 from FADH2 and 1 from GTP. This is a simple math calculation I think you will be able to do, and I'll tell you how one NADH is producing 2 ATP in the electron transport chain. For now you remember this. We will discuss that in detail in the electron transport chain, how one NADH is producing 2.5 ATP and one FADH2 is producing 1.5 ATP. So what are the metabolic applications of TCA cycle? It is involved in lipid metabolism, amino acid metabolism, protein metabolism and turnover, glucose metabolism and energy expenditure. So regulation of TCA cycle, very important metabolites. The NADH which is produced in the TCA cycle can inhibit major enzymes. Okay, so you see the product can inhibit the enzyme. Here we call it as negative feedback. Next, citrate. Citrate is a important compound that can inhibit the phosphofructokinase in the glycolysis. Okay, it can inhibit the phosphofructokinase in the glycolysis. Also, this reduces the rate of production of pyruvate and therefore of acetyl-CoA. Okay, calcium can accelerate the TCA cycle by stimulating the link reaction. Okay, so these are the regulations. Remember these regulations because based on the regulations, they'll definitely ask you questions. I hope you enjoyed the class of TCA cycle today with me. So you can subscribe to our YouTube channel, Gurmantra. Comment down below in the comment section how you like this class. Like, share and subscribe to our YouTube channel, Gurmantra. You can join our Telegram channel, Facebook page, website. And if you have any queries, you can mail us to tanejanehaofficial@gmail.com. You can contact us on 9351280000. Thank you so much. Have a nice day. So the mantra, always remember the mantra. What is the mantra? Study, revise, practice and repeat.